technical issues being worked out. I think we're ready to go. Thank you all for coming. This is the last seminar event for the BIMR of the year. So thank you. I've had a great program, and it's a real pleasure to finish the program with the event today, which is the presentations of the two groups of graduate students that were successful in the Future Materials Innovators program that the BIMR has put on now for this year is the third year in a row. And this is the second group of students that have received this award. And for those that aren't aware, this is a program that the Broncos Institute launched a couple of years ago. We're very interested in promoting, encouraging, and supporting ideas by graduate students for research, and in particular collaborative research projects between multiple groups, preferably in different departments and more preferably in different faculties, so that they get together and work on a project that kind of merges expertise between multiple groups. So this is one of the mandates of the BIMR, is to promote interdisciplinary collaboration. So it gives me great pleasure to introduce the first of our two groups for today. And the first group is composed of Brian Singer and David Gonzalez Martinez, along with a couple of collaborators that are no longer in the group. And they're going to present their work on the development of a mixer for homogeneous, reproducible, and gentle mixing of cells and viscous filings based on silk fibrine methacrylate. So, take it away. Thank you.
Thank you. Yeah, so we're going to tell you guys about this mixer we've been developing for mixing cells into viscous bioinks, such as this new material, silk fibrine methacrylate. Note Mabel and Stephen can't be with us. They've moved to other institutions, but they had large parts to play in this project in the initial stages. I always have issues with this. Here we go. Okay. So extrusion-based 3D bioprinting is becoming more and more important in biomedical research, mostly for its ability to create custom cell-laden 3D biostructures. And this is important for tissue engineering and regenerative medicine applications and microphysiological systems, among others. One problem currently with extrusion bioprinting is the bioinks, or the materials that are used in extrusion bioprinting. They have high viscosity, and this makes it difficult to mix with cells. So if you want to try and keep the cells happy, you may not get a very homogeneous mixture. And if you really try and mix them in there, you might get low cell viability. So there's a need for a simple and reproducible method for mixing cells gently within viscous bioinks. The project was divided into two major objectives. The first being to develop and characterize a bioink based on silk fibrine methacrylate, or silkma. And David will walk you through the composition, rheology, and printability of this material. And the second major objective was to develop a static mixer for incorporating cells within silkma and other viscous bioinks. And we'll show you a few different geometries of mixtures that we tested. We'll show you some computational fluid dynamics, or CFD simulations, to show shear stress. We'll show you the actual physical performance of the mixers, and we'll show you the viability of cells after mixing.
Thank you, Ryan. So, as Ryan mentioned, the first step of this project was developing a silkma, silk fibrine methacrylate bioink, for extrusion 3D bioprinting. It's important to notice or to mention that when you're developing inks for extrusion bioprinting, one of the main characteristics that you need to take into account is the viscosity of the ink. For example, if you try to extrude this silkma without any rheology modifier, it's going to be almost impossible because you're going to have a liquid that flows and the printability is going to be too low. So basically, you need to add a rheology modifier to enhance or improve those rheological properties, right? So the first step of this part was synthesizing the silma. To do that, we extracted the silk from the silkworm cocoons, and then we modified using glycidyl methacrylate, a reaction where mainly the primary amine groups of the proteins react. They react with the epoxide groups, and then we have the double bonds here that would react with the UV light and a photo initiator for the photo cross-linking reaction during the bioprinting. So after synthesizing and getting the Sigma, we design an workflow in order to have our bio inks or biomaterial inks. First, here we have lab is the photo initiator that we use. Carbapol is the rheology modifier that we have been using at Moran Mirabal Group. We have proved that this rheology modifier can be used for different proteins or polymers, synthetic polymers that cannot be extruded by themselves. And this rheology modifier enhance those the rheological properties of those polymers and allows the bioprinting of the polymer with a high printability and reproducibility. So we mix the Silma, the rheology modifier, and the lab. Then we put all those in a syringe. We mix with 2 syringes. Then we centrifuge to take out all the bubbles, and then we have our biomaterial ink. Important to mention that the main difference between biomaterial inks and bioinks is that this ink doesn't have cells inside, so we call this biomaterial ink. So when we are developing inks for extrusion 3D bioprinting, we need to look for 2 main properties other than just viscosity. The first one is shear thinning, and we analyze 3 different formulations. We analyze Sigma 1, 3, and 5% with 0.75% of Carbapol. And we can see here that when we increase the shear rate, which is proportional to the shear stress, the viscosity decreases. So basically that's shear thinning. And why is this important? Because this is important because imagine that you have your ink inside the syringe. When you put that pressure, you're putting some shear stress, and then that stress decreases, and the viscosity decreases, and you can extrude your cells without damaging. You can keep a high cell viability. And second, we analyze the thixotropy behavior of the formulations. This is like a simulation of the printing process. Here we have a low shear rate. The viscosity is stable, it's constant. This would be instead when the ink is inside the syringe, for example, then you suddenly increase the shear rate. That would be the extrusion process, and then you take it back to the original shear rate. That would be after extrusion, the filament. So as you can see, we have a perfect thixotropy behavior with a good recovery, which implies that we should have a nice or a good printability using these materials. After that, we analyze, we just photo cross-link using the photo initiator that we mentioned before. We photo cross-link the inks, and we analyze the mechanical properties. As you can see here, we have G prime, G double prime, loss storage modules, and the compressive modules. And this is a log scale here. As you can see, we increasing the concentration of the main polymer, the protein. We can enhance or increase the mechanical properties of the hydrogels that we obtain. And after that, we selected the 5%, the 3% formulation, and we print that biomaterial ink. We try 2 main models. The first one is a cylinder, and the second one is a mesh-like construct, which is typically used for proving or demonstrating high printability. As you can see here, in both cases, we have a good shape fidelity and a high printability. These squares here are quite similar to the model that we tried to print. And something that was really interesting for us was that when we tried to stretch this same cylinder, we obtained this kind of behavior, super stretchable hydrogels, which is something remarkable. Sorry.
Now onto the mixer portion of the project. Can you guys at the back hear us? I realized that we weren't using the mic, but you can hear us? Okay, sweet. So we evaluated 3 different geometry types of mixers. One is a serpentine like design where the fluid flow is split and then goes through 2 90-degree turns and then is recombined, and that happens over each element. Another is an SMX type design where there are these X structures, these grids that span the width of the channel, and with each element they're rotated 90 degrees. And then there is a Kenix style mixer, which has these rotating elements that change direction of rotation with each element. So these were printed using a 3D resin printer. And so you can see the scale of these. That's roughly how big a quarter is next to these 3D prints. So the resolution of these is quite fine. Like for the serpentine model, those are like 1 by 1 millimeter, and we're able to get that very reliably. So we're going to show you some fluid dynamic simulations of the shear stress that we expect cells to experience in these mixers. And we're also going to show you the mixing of fluorescent particles into PEG-DA. So for the CFD simulations, we used Autodesk CFD. And for the fluid properties of Silkma, we actually interpolated experimental data to obtain a power law relationship. And that's what we inputted into Autodesk CFD so that we could get a good idea of how this fluid is going to behave. For the turbulence model, we use a standard K epsilon, and we computed solutions to steady state. So the reason that we're interested in looking at shear stress in the first place is because we want to keep the cells happy, and higher shear stress damages cells and can reduce your viability. So these data we did not generate. This is from that source up there, but it's showing that once you start getting to shear stresses of 5 kilopascals and above, you start getting significant losses in cell viability. So we set that as one of our design constraints, that we need to keep shear stress below 5 kilopascals. One thing to note about the software that we used is it's incapable of performing mixing analyses in which one fluid has the complex rheology that Silkma does, where it's interacting with another fluid with different rheology. The simulations just can't happen in this software. So we had to choose one of these fluids to simulate flowing through our mixer geometries. And we found that at the same flow rate, flowing Silkma only through is the determinant of maximum shear stress. So for the simulations that I'm about to show you, we're showing Silkma flowing through the mixers. So looking at the Kenix style mixer, what you're looking at is the fluid volume within the mixer. And the color is showing the shear stress, with blue being low shear stress and red being 5 kilopascals. So at both a low flow rate of 1.25 and a high flow rate of 6.25 milliliters per second, the shear stress is not approaching 5 kilopascals. So this is probably going to be suitable for keeping cells alive and happy. Even more so with the SMX style mixer, even at a high flow rate of 6.25 milliliters per second, which is very fast when you think about, like, say, a 50 milliliter syringe perfusing at that rate. That's very fast. You probably wouldn't even use that rate. But even at that flow rate, we're not approaching 5 kilopascals at all. So this will probably keep cells happy. Then looking at the serpentine mixer, when we get to a high flow rate, there's a chance that it might hurt the cells. So this is still an option if it's a good mixer, but we would have to keep the flow rate low to keep the cells happy. So now looking at the actual physical performance of the mixers, we mixed fluorescent particle suspensions with PEG-DA. And the reason that we used PEG-DA was because it's more accessible than Silkma, and it has similar rheological properties and density to Silkma when it has the same additives, carbapol and lab. So we use syringe pumps to control the flow rates of both of these liquids at a 4-to-1 ratio, and they combine together in a Y channel. They flow through the mixer and into an output syringe. And then we cure that output syringe with light to cross-link it, and we cut it open to get that cylinder out, and then we slice that cylinder to get fluorescence images like that in the top right corner. So we upload those images into image J, and we take a region of interest across the entire slice, and we simply take the ratio of the standard deviation of pixel intensity over the mean pixel intensity. And this gives us a single number, which is the coefficient of variation, that tells us how homogeneous the pixel intensity is inside that region of interest. So you can imagine if it was all one color and one intensity, the variation would be 0. The standard deviation would be 0. So that would be best case scenario, the coefficient of variation of 0. And as you get less homogeneous, more heterogeneous, you get a higher coefficient of variation. So what you're looking at here is on the bottom are the numerical values for the coefficient of variation, and then qualitatively are representative slices. And we're comparing each of them to, or sort of using a complete mixing scenario as a benchmark. So that's us really mixing these fluorescent particles into it to make sure that they're completely mixed, and then taking a slice like we would with these. Notice that the coefficient of variation for this complete mixing scenario is not 0. It's not quite 0 because of artifacts in the image like lines from slicing, air bubbles, other things like that. And those are present in our real images too. But it gives us something to compare to. So with one pass through the mixers, meaning that the material was passed through the mixer once into the output syringe and then measured from there, we can see that the coefficients of variation are well above 1. So the standard deviation is much higher than the mean pixel intensity itself. So this is very not homogeneous. In these black regions, there would be no cells, and that's not good for printing. So if we pass it back through the mixer, which is essentially the same as doubling the length of the mixer too, we see that we start to get lower coefficients of variation, and visually it looks more homogeneous, especially with the SMX style. But still there are some dark regions that would have less cells than the brighter regions. So if we pass it through another time, we can see that the SMX is now getting quite a homogeneous distribution with a coefficient of variation that's well below 1. And so with that paired with the fact that the shear stress was very low for the SMX model, we chose that to test with cells. So these are 3T3 fibroblasts that are being mixed into Silkma to make a bioink with around 5 million cells per milliliter. And then we used live dead stain to look at the live cells in the green channel, the center, which should be red, but there are very little dead cells. And then those two channels merged on the right. So the two things to note about this are very high cell viability, low cell death, and qualitatively, it appears that the cells are quite well dispersed within the bioink. Another thing that we noticed is that there's a bunch of little specks or dots in these images, and those aren't cells. So the larger circles are cells, but those little specks are not, and this could be autofluorescence or some other phenomenon. It's just something interesting that we noticed. So to conclude, we developed and characterized a bioink based on silk fiber and methacrylate, and we showed the printability and strength of this material. We also evaluated 3 different styles of mixers, from which we identified the SMX as having the lowest shear stress and the best mixing performance. And we showed that the viability of 3T3 cells was high after mixing with the SMX. So thank you very much to the BIMR Institute for providing us the opportunity to do this work, and thank you to our supervisors who let us know of it, and thank you all for listening.
Okay, we have time for a couple of questions.
Thank you so much for the great talk. I was wondering with your cell study, when you have it, if you have it cured, how did you go about controlling the viability of the cells after
That's an excellent question. Actually, this experiment was without curing because the material is highly viscous. What we did was just extrude small dots in a well plate and then just add directly the live data side and without any UV light and then see the viability. Actually, we did both, but we realized that we could do it without UV and then we're just presenting that data because we are interested in the viability after mixing, not the viability after mixing plus curing, right?
So the question was, are we going to try with different sizes of cells and different types of cells? The answer is yes, we do. We do want to do that. We were going to use epithelial cells as well as fibroblasts, but we only ended up doing fibroblasts at this point. But I think it definitely is worth testing different sizes of cells, types of cells, maybe some that are not as hardy as fibroblasts would be worth looking into.
Simply curious about the sources of some of your materials. So in particular, all the mixers, are those made in-house or are those commercially available? And also for soap, is that also bought commercially or perhaps bring soap firms into that?
I guess I'll start.
Yeah. Go ahead.
I'll start with the mixer materials. So you're asking where we source materials from, like the mixers and.
The mixer in particular, is that made in lab or is that commercially?
So the designs were inspired from mixers that already exist. So the names of those mixers already exist. Actually, the SMX, I don't know what that stands for. I think it's the company Solzer. I think that's a trademark by them, but I couldn't find what it stood for. Anyways, so the geometries, they already exist, but these are ones that we modeled based off of them. And we printed them in-house using these resin printers. And then for the
Yeah, and for the materials for the bioinks, we got the cocoons and then we extract the seals by ourselves.
Nice, we should probably bugs into lab. Let me know next time.
You mentioned that with mixers, you can either pass back and forth, but that's the equivalent of doubling the length or tripling the length. So which of those is better, to pass back and forth or just have a longer mixer that you just pass through once?
Go ahead.
Yeah, I think that's an excellent question. Yeah, both have advantages and disadvantages. The main one is that okay, if you have a mixer that is too long, then you're gonna have a large dead volume, right? But then if you go back and forward, you can introduce a lot of bubbles. So for this experiment, we decided to go with a larger one. Just we printed for the experiment with cells. I mean, since we learned that we need like 3 passes to have something that is completely mixed, what we did was just print a larger mixer and then use it, right? So I think that the best idea is using the larger one.
Might also be better for automations.
For automation, yeah, definitely.
Certainly.
So, thank you for this very interesting talk. So, I'm missing that what should be the ultimate use of this cell impregnated silk mark that you want to use for as a artificial organ or tissue, something like that?
Yeah, actually, Sigma is a protein base. I mean, it's a protein, and then with this kind of protein, we have in our lab we have used all the kind of protein for making tissue ECM biomimetics for tissue models, for example. And Sigma is a potential candidate for printing different tissues. I don't know if you remember one of the slides we mentioned the mechanical properties, and those mechanical properties are close to several organs, right? So ideally, we should try to test different cells from different organs and see the behavior of those cells.
But the the cell can proliferate on this matrix sigma.
We haven't proved that.
And whenever you have done this viability studies, did you, the viability which is being impregnated, but you started with number of cells, and which one, how many cells are making impregnated, and during this mixing, how many cells are becoming dead? Did you calculate the dead cell versus live cell which is being impregnated over there?
So that would be viability, right? Live versus dead cells. So yeah, we
The yield, biological yield.
yeah we didn't calculate. I mean, the number of cells. We know that we put 5 million cells per milliliter, but we didn't calculate the number of cells that we have at the end. We just got the viability of the some sample of the ink.
Great, okay, I think we should move on to the second talk, so please join me in thanking the first. Okay, so our second presentation is going to be given by Michael Solozowski, Andy Marway, and Anthony D'Angelo. And the talk is entitled Modeling Drug Clearance Using iFlow Plate. And I should point out that these are collaborations. So the first group was a collaboration between the Mirau group and the Rhoda group. And this is a collaboration between Zhang and Wiley groups.
Hi, everyone. My name is Mandeep Marway, and I, along with my colleagues, Michael and Anthony, will be discussing our BIMR project focusing on modeling drug clearance using the iFlow plate T.
When studying cancer in vitro, we can look at cancer cells that are adhered along the bottom of a plate. This is a 2D cancer monolayer, and essentially you have drug suspended in media that's above it. And while this isn't too bad for studying drug binding and action, the question is, how does it differ in the actual tumor environment? This is where you have cancer cells that are in a 3D form, and there might be other biological factors that are contributing to any drug activity. The other issue is that we don't see continuous clearance in this model. Typically, you would have drug that is physically removed from the local tumor site, and you also have drug metabolism. So you're not seeing the exact same effect that you would see biologically. Another factor to consider is that the tumor environment is one of high pressure. And what this means is that for especially smaller drugs, you have less retention of the drugs in the environment. Biological pressure gradients do exist, and in the case of cancer, we're talking about a high pressure tumor environment and a region of less pressure surrounding that tumor environment. So ultimately, we have lower drug retention. We can use local sustained release technologies to localize our therapeutics, such as hydrogels. And what this allows us to do is increase the therapeutic half-life for those challenging therapeutics locally within the tumor site. And we can augment the dose within the gel. And this allows us to hopefully improve efficacy and hopefully circumvent any systemic toxicities.
Okay, so now that we know that we have both the pressure gradient in the cancer environment and we need local delivery systems, allow me to introduce the iFlow plate T. The iFlow plate T is a microfluidic device that's created on a 384 well plate that contains 64 independent networks. Each of these networks are created by connecting 4 wells together. So to mimic the high to low pressure gradient, we begin by creating a high pressure environment in the 3 adjacent wells that are connected with a single channel, and a low pressure gradient, a low pressure well in the clearance well that's attached to the center well. The center well will be used for setting up our tissue culture. Okay, so in order to check whether or not our pressure gradient actually works, we used dextran as our model molecule. So we began by passing a empty fibrin gel within the center well and then adding dextran on top in order to mimic intravenous injection. And what we found was after 24 hours, we noticed that majority of the dextran was being cleared out into that clearance well, that fourth well that's attached to that center well, compared to the 2 outer wells. So we know that our pressure gradient is working. However, a lot of the drugs that we want to test need to be administered locally using a vehicle. So for that reason, we decided to use agarose. So the reason why we need to use some sort of sustained release or some sort of delivery vehicle is because a lot of them are toxic when administered intravenously. So again, we began by adding the dextran within the agarose gel. We used 2 different molecular weights of dextran, both 4 kilodalton and 65 kilodalton. And what we found, again, using the high to low pressure gradient, was that we did have dextran release into that clearance well after 24 and 48 hours. And we also noticed that the 4 kilodalton dextran was actually being released more rapidly than the 65 kilodalton dextran, most likely due to the fact that it is smaller. So while dextran is a good representative of what a potential drug would show, we wanted to move forward with using a real drug. So in order to set this up, we began by adding our drug within the agarose gel, and then casting cancer spheroids. We're using glioblastoma as our model, but it could be used for any cancer. We casted cancer spheroids within a fibrin gel, and then added an endothelial monolayer on top with monocytes on top of that. So monocytes are immune cells that are able to flow through the bloodstream, and upon detecting cytokines or signaling molecules that signal for the monocyte recruitment, they then bind to the endothelial monolayer and pass through to the tumor site. Previous work that we've done in the lab shows that the presence of the endothelial monolayer is necessary for the differentiation of monocytes to macrophages, which are our target immune cells. Additionally, we could also differentiate these monocytes with chemicals such as PMA. However, they do lead to phenotypic and genetic changes, which could reduce our biological relevance.
So as Mandeep mentioned, these monocytes are going to differentiate into cells called macrophages. And then these macrophages are going to interact with the cancer in a way where they both benefit. So the macrophages are releasing these anti-inflammatory molecules that are helping the cancer to grow and survive. The cancer is then going to release some signaling molecules, and this is just going to help recruit some more macrophages. So in our system, we want to attempt to target this. So we're going to use this model drug combination. So the 2 model drugs are interferon gamma, which is a type of cytokine, as well as an antibody. So the antibody that we're picking, it's going to bind to a specific marker on the glioblastoma cells known as CD133. So we'll just call the antibody anti-CD133. The interferon gamma, it changes the macrophages in a way where they're not going to release so much of these anti-inflammatory molecules anymore, and it helps to break up this crosstalk. But then the anti-CD133, what this can do is it can actually bind to the cancer cells, and it can actually mark them, that mark to the body that the cancer cells that they need to be cleared out. And the macrophages, provided they're polarized, they can actually help clear the cancer cells out. So we did a bit of preliminary work with a breast cancer cell line just to show this type of model treatment. So we did test interferon gamma, but we needed just a very slightly different antibody. So I had mentioned CD133 for glioblastoma, but there's just a different marker on these cells known as HER2. So we'll use the anti-HER2 antibody for this study. So what you can see is we measured the cell viability with different drug treatments. And when we try these two therapies on their own, we only see a modest reduction in cancer cell viability. But what we can see is that the viability actually drops by a factor of 2 with the combination treatment of both an antibody targeting the cancer cells as well as with this interferon gamma cytokine.
So we've shown the capacity to target breast cancer cells using a combination of IFN gamma and anti-HER2. Now moving towards brain cancer, glioblastoma, or GBM stem cells. Now we're moving towards the anti-CD133. And typically these cancer stem cells, they're cultured in neurocult media, which doesn't have fetal bovine serum. But in the case of the ifloplate, we're moving towards cell culture conditions that contain EGM2, which has 2% FBS. So what we want to ensure is that it doesn't cause a differentiation of these cancer stem cells, causing it to potentially lose the marker that we're interested in. And what we see here is through flow cytometry, we see that the expression of CD133 is negligibly affected from the one media to the other. So going from 10% to approximately 8%, our baseline level of CD133 is retained. So the hypothesis for this work with the ifloplate is that differential tumor spheroid killing will be observed with a combination therapy of anti-CD133 as well as IFN gamma. And this will be under the clearance model. Local sustained release of IFN gamma using the agarose gel should allow us to enhance killing compared to the administration of IFN gamma just locally within the fibrin gel. And the reason for this is that IFN gamma is one of those therapeutics that are challenging. They're smaller and they are not as easily retained in the tumor environment.
Okay, so we know from previous work that the combination therapy works, but would this work against glioblastoma? So we began by first looking at the native environment. So we wanted to see if there was a reduction in spheroid size measured using the fluorescence emitted by these spheroids. So essentially the way that I measured this was I measured the amount of fluorescence or the area of fluorescence emitted by each spheroid over time, and then divided by the number of spheroids within that well. And what we found was that when no therapy was used, there was no significant change in the spheroid size or the spheroid viability. And additionally, when only the CD133 was added, there was also no significant change. Similarly, when we added just the IFN gamma, again, we didn't see any significant change in spheroid viability. However, when we did have the combination therapy, when we just administered the IFN gamma without the agarose gel, along with the anti-CD133, we did see a significant reduction in spheroid viability. However, when the agarose was present with the same concentration of IFN gamma added, we didn't see that same trend. So what we're suspecting is that the amount of IFN gamma actually being released from the agarose is not sufficient to reach the therapeutic window. So further studies are necessary in order to optimize our initial concentration for the agarose, or for the IFN gamma in the agarose. So the next thing was we wanted to know how much IFN gamma is actually being released from our system. And so for this, we decided to do a cytokine analysis. However, it's going to be a couple of weeks before we get this data. So we're still waiting on it. But based on previous work that has been done, we had a similar setup without the IFN gamma actually being administered into our system, just to look at the native environment with the endothelial monolayer, with the spheroids and the monocytes. And we saw that our baseline IFN gamma expression is very low, and our environment is more pro-tumor. So it seems that the macrophages that we're getting are actually promoting tumor growth. However, what we're expecting is that with the administration of IFN gamma, we'll have more of the pro-inflammatory or anti-tumor cytokines being expressed, such as IL-12P70, which would help with killing.
So using the iFlow Plate T, we have a platform to model continuous clearance. We can capture features of a tumor environment. Based on the previous slide, we saw the more pro-tumor environment with the unique cytokine profile there. And together with these two factors, we have the means to study drug action under more biologically relevant conditions.
So in this study, we used one specific antibody, anti-CD133, and one specific cytokine, interferon gamma. But these aren't the only two drugs that are put under investigation for potentially being used in glioblastoma. So the idea is we want to use this platform in future work as a drug screening tool, so we could test different drugs or different drug combinations against a single biological model. So for example, in this plate, there's actually 64 networks in one plate, so you can actually screen multiple drugs right on a single plate. But in terms of biological models, we're not limited to just targeting macrophages. There's other immune cell types that are involved in glioblastoma, such as T cells. So this could be used to change the biological model, and then you could screen for drugs involving T cells. But again, this isn't even necessarily limited to just immune cells or glioblastoma. You could change the disease model to other types of cancers, or even broader, you could change to completely other disease models, such as fibrosis.
And with that, we'd like to thank the BIMR Institute, specifically Dr. Adranoff and Florian, for helping with getting the cytokine analysis done. And our PIs, Dr. Blaine Zhang and Dr. Ryan Wiley. And a special thank you to Kevin Wei for helping us with the flow cytometry, and Sahib Marway, who helped with the manufacturing of the devices. Thank you.
Questions?
I wanted to ask, one of the points you made that you want to be able to test several different drugs using this, so that means it's inevitably amenable towards high screening automation. Have you tested this at all in this machine that the John Lab has for high screening yet? Or?
No, so we haven't reached the point. Well, okay. Technically I can, but we don't need it to that capacity yet, but it can be applied. It would probably be better to use the Nimbus at this point, just to reduce variability that's from person to person. But as it stands, we haven't tested it yet.
And also a supplemental, how small are those, what are the volumes of those again?
So the gel, the fibrin gel on its own is 15 microliters. And then the agarose was 10 microliters. Total volume in the well was 9. So topped up with media. Yeah.
Use a restraint a little bit.
Yeah.
Thank you.
Especially for media changes.
So yeah, thank you for this interesting talk. I was wondering that agarose gel to the IFN gamma is acting as a stabilizer or delivering agent to the cancer cell.
It's a delivering agent.
Delivering agent to the macrophages or the cancer cell.
So it's just delivering it to the tumor microenvironment, but in reality, we're not going to inject agarose into a patient. We just in the lab, just for preliminary testing, we wanted something relatively simple.
No, my point is like whether you know the mechanism of action, because IFN gamma is abundant cytokines. It is an anti-inflammatory marker. Is already there in our body. So what is the additional stuff that you are giving in order to trigger the cancer cell mortality? And it is like a apoptosis or is a necrotic pathway that you are trying to get?
Well, it's targeting the macrophages. So you want to turn the macrophages, they're secreting anti-inflammatory cytokines. So interferon gamma, it's going to act on the macrophages and it's going to create a pro-inflammatory environment. So it's going to start all these other pathways. So for example, the macrophages, they're going to start secreting IL-12. This is going to help bring T cells into the environment. This is going to start killing the cancer cells. So the idea is it's just about like polarizing the environment, but then you have the antibody coming in, and this is actually marking the cells. So then you can start potentially getting phagocytosis or ADCP, you can get ADCC. So it works as a combination type treatment. And another reason too we want to do it local is because, yeah, it is pretty toxic. You know, it's going to go, it's going to cause a number if you were to just inject it into a person. So that's why we need the aggros, so that way we're delivering it right to the site of action.
And like in the body, if we were to administer it, like in a hydrogel, what typically happens is that most of the tumor is surgically removed, and that leaves the cavity there. So we would be injecting it within that cavity, so that it'll secrete it, act on the immune system as a whole, not necessarily just macrophages, but on the immune system as a whole, and then promote them to actually go and kill the cancer. So yeah, so it would be on the macrophages more so than the tumor itself.
So in that case, you have to be careful about this mode of injection. Like you will just give the intraperitoneal or you are giving the oral, something like that, because your injection will depend upon the mode of administration of the drug.
Well, it would be local delivery. That's why we, yeah, so that's why we put in a hydrogel. It would be in an injectable hydrogel that we can actually put like directly into the cavity after removing most of the tumor. Because when you try to surgically remove a tumor, especially a brain tumor, you can't get rid of all of it, because as you try to remove it, you're also potentially losing functionality of the person. So you want to try to remove as much as you can without destroying the person, and then injecting the drug in that location
Okay, maybe
so that it
it's
can
up
further.
there. This
Yeah,
is
yeah,
how
we'll
good.
discuss,
Yeah.
Other questions are waiting.
Yeah, I have a technical question about your 356 well plate. Is there a reason why you're having always these 2 free wells? You didn't have a good Tetris player in the lab?
I actually, so for that, when we were designing the plate, we thought about that. But in order to, there was a couple of things that we could do. So instead of having a T design, we could actually connect the 2 empty wells as well, to that clearance well and just create like a larger reservoir. That can also affect our flow rate a little bit too, by increasing it. As far as like trying to like shape it together so that we use all of the wells but keep the T, it'll be a bit hard to actually work with at that point. It just becomes too complex. But yeah, we tried the Tetris design that wasn't going to be viable for working with. But
So I wanted to ask you about the pressures. You mentioned the difference in pressure between the tumor environment and the outside. So I was wondering, when you design your teeth plate, what are the pressures that you can achieve with that system, given that it's gravimetrically controlled, if I understand correctly? And how we compare it to what you have?
So we didn't do any particular testing of what the pressure is. But as far as mimicking the tumor environment exactly, I think it would be too small of a scale to get exact values to mimic what happens in people. We're just trying to get like a like just model the general like high to low. And we can actually adjust the pressure as well, through like different methods, either by changing the ECM, by changing the volumes that we work with. So all of that can be adjusted. We just haven't tested exactly what that pressure is.
So you basically adjust the pressure by how fine the mesh you have, and you push it through.
Yeah.
But not the trigger pressure.
before we thank both groups again, I just want to remind you, there's a reception just outside here in the foyer, so don't run away with some food and drinks. And the two groups, I'd like to get photos, so don't run away until we do that. And with that, please join me in thanking the second group of
Interesting, though. I don't know what we